Aloha, this is Dr. Tom coming back at you. We're talking about the use of lipids. Lipids are fats, very important in the body. Why? Number one, they're a source of energy. Now, in modern times, that may not be as much of a problem in industrialized countries, but years ago, when people didn't know where their next meal was going to come from, fat was real important because it has twice the calories that proteins and carbohydrates do. Also, lipids are an essential part of the cell membrane, and we spend a lot of time talking about how important that is. Also, some of our nerves, many of them, have a fatty whitish coating around it called myelin. Cholesterol and fats play a big role in that. Also, cholesterol is one of the lipids extensively seen in the cell membrane. Steroid hormones, which are important to the body, have a lipid component to them. It's real important, and long-term storage of energy. So fats are very important. Well, all three of the foods are. Now, let's talk about, you've all heard of this probably, good and bad cholesterol. The fattier it is, the worse it is. Fat is not very dense. That's why oil will float on water. So low density lipoproteins or cholesterol or fatty acids, triglycerides, I'll get it right here in a minute, are the bad ones, and the high density are the good ones, okay? So the worst of all is the very, very low density lipoprotein, VLDLs. Why? Because it's mainly fats. Transports triglycerides to adipose tissue and muscle. Adipose tissue is the fat we have stored on our body, like the beer guts and the love handles and the triple chins and that kind of thing. So the worst is this one. The next worst is LDLs, low density lipoproteins. Why? Because it has more cholesterol and fat. Deliver cholesterol to non-hepatic tissue. Now, what's non-hepatic tissue? Well, hepatic means the liver, so it would mean anything but the liver. The healthier cholesterol are the HDLs. And it's commonly just called by these initials, guys, so you need to learn those. It has less fat and more cholesterol. It's healthier. Carry cholesterol from peripheral tissues to liver for excretion. Okay, so foods with more HDLs are healthier than foods with more LDLs or VLDLs. Okay, and that, in a nutshell, guys, is good and bad cholesterol. Cholesterol is absolutely essential for life. It's in myelin sheath, all the things we talked about a moment ago. Steroid hormones, cell membrane. So it's been made out to be a terrible thing, and it's not. Most of our serum cholesterol, the cholesterol that is measured in our blood, the bulk of it is produced by the liver. Only a relatively small amount of it is due to dietary and lifestyle, but some of it definitely is. Okay, proteins. The third food, most abundant organic matter in the body. Why? It's all the structural stuff. Proteins participate in every body function as enzymes. We talked about how important enzymes are. They're the catalysts of the biological world. Hormones. Hemoglobin, the molecule that transports oxygen and carbon dioxide in red blood cells. Contractile muscle proteins. Plasma proteins, important in the blood. Antibodies play a huge role in immune function. And various structural proteins in the body. Now, the building blocks of proteins are called amino acids. This is very important, relatively simple. Amino acids are the building blocks of protein. You need to know that. All amino acids have an amine group and an acid group, which is why they're called. They're joined by peptide bonds. Anytime you see the word peptide bond used, they're talking about a protein. Also, little small fragments of proteins are called peptides. So here's the amine group, the acid group forming the amino acid. Here's one, alanine is one amino acid. These are carbon molecules here, hydrogen, nitrogen. Proteins, guys, are distinguished by the fact that they have nitrogen in them. So peptides are little tiny proteins. Peptide bonds form here, and the full polypeptide is a protein. Okay, now some amino acids are essential, which means we have to get them from the diet, and some of them are non-essential, which means we can make them from the essential ones if we have them. So the essential ones we absolutely must get in the diet. Non-essential, the body can make them if we have enough of the essential ones. Of course, the liver does that. The liver is amazing. It has over 400 functions. Both are needed by the body. Use of proteins, synthesis of hormones, enzymes, antibodies, plasma, muscle proteins, hemoglobin, most cell membranes. We already mentioned all that. If needed, we can burn proteins for fuel to make ATP. They can be broken down as a source of ATP production. If needed, they can be broken down and converted to glucose via gluconeogenesis. Now, the problem is that proteins have nitrogen, and nitrogen builds up in the body and becomes toxic in a sense. And so we have to eliminate it from the body. So nitrogen is a waste product of the amino acid breakdown. Proteins are distinguished by the fact that they have nitrogen. Excess nitrogen forms ammonia, which we all know ammonia is how rough that stuff is. Very caustic, very high, very high in the pH scale. The liver removes toxic ammonia from the blood, here's the liver, and converts it to a substance called urea. The urea, in turn, is transported to the kidney, and the kidney excretes urea in the urine. So there's a complex procedure here, guys, in how to handle all this stuff. That's how important it is. Okay, so the excess nitrogen is converted to urea. See, we're getting closer to the word urine, see. It's transported onto the kidney, and the kidney has a variety of ways of excreting, or, well, some processes to go through to excrete the urea in the urine. Okay, we all have heard of DNA. There's also its first cousin, RNA, which is a simpler form of it with a different function. The DNA molecule, when it was discovered by Watson and Crick years ago, guys, was a major, major discovery. And they found that it was in the form of a double helix. If you took a ladder, like a long ladder, and you bent it in a spiral, where it was like that, that's the shape of a DNA molecule. And the rungs of the ladder are made of these nucleotides, adenine, thymine, guanine, and cytosine. Sugar base, and then that. The nucleic acids are DNA and RNA, composed of nucleotides. What are the nucleotides? Adenine, thymine, guanine, and cytosine, and bacteria has one called uracil. Okay, these things pair only in certain ways. And that's what we call by base pairing. DNA is a double-stranded nucleotide, like a twisted ladder. The rungs of the DNA ladder are formed by base pairing. Of those 4 nucleotides, adenine bonds with thymine, and cytosine bonds with guanine. Adenine bonds with thymine, cytosine bonds with guanine. Do you need to know that? Yeah, at least enough to get through the test on it. Okay, here's a drawing of the DNA molecule. See how it looks like a twisted ladder? The rungs of the ladder are these nucleotides, and here they are. So thymine bonds with adenine, and guanine and cytosine. And so that's what these are. It's just repetitious structures of these bonding in that certain way. Okay? Now this is important. May not seem like it at the moment. Because the way these things bond and the way you read the body reads them out is what contains the genetic code. Genetic information for protein synthesis is stored on one strand. The order of 3 bases code for 1 amino acid. Okay, so the way these, once they're bonded, the order that they're in determines an amino acid. So these things are red. They're opened up in red, or if they're in, I mentioned earlier in another video, chromatin for the DNA is like threads. The RNA can go in there and read it then and tell, but these things code for amino acids, how to assemble a protein. The order of 3 bases code for 1 amino acid. So RNA, we're going to talk a little bit more about it here in a moment, goes down through this and reads this. Here's the nucleus. See, this was contained in the nucleus. And these 3 codes, like this one here, codes for a single amino acid. This one codes for another amino acid. And this guys, is sort of like not a binary, but yeah, well, maybe almost would be. Anyway, it's the instructions on how to build and put together a protein. This, boys and girls, is what the genetic code tells. Now, I know what you're thinking. You're thinking, Dr. Tom, that's not enough. You're right. Knowing the amino acid sequence of a protein leaves out a bunch of stuff. How is the rest of the molecule formed? Because organic molecules often are a million, it can be a million carbon. Well, they can be many carbons long, and they twist in certain ways. And if they twist the other way, they can't be used. And it's called, and they can be in different shapes of the same exact formula, and that's called stereoisometry. Or if they're twisted in one way, which is called chirality. For example, glucose. The form that's used throughout the world, all animals and stuff, is L-glucose, has a left-hand twist. Right, our glucose cannot be utilized by living things, or at least for the most part. If there can be, it's something I don't know about. Where's all that in here? Well, I'm going to tell you a little hint. It's not there. We don't know where it is. But that's just the tip of the iceberg, guys. Because what else we don't know? This just codes for a protein. Where's the instructions on how things are shaped and where things move and how arms turn out to be arms and eyeballs turn out to be eyeballs? We don't know. It's not there. This is the biggest mystery in biology and has been for quite a while. And they are scrambling to come up with answers. We don't know. We only know a tiny bit of it, guys, when it comes to genetics. And also, a lot of animals and plants have almost identical DNA. I wish I could remember the exact figure. I started to look it up, and I didn't have time. But there's only a fairly small difference in the DNA of a rice plant than the DNA of a human. So there's some major stuff missing here, guys. I've been looking into this. I'm interested in this. I've looked at it for quite a number of years. The stuff that I tend to believe is not supported by mainstream science, but so what? As time goes on, we find more and more their stuff is often wrong. Okay. Now, we mentioned RNA. There's messenger RNA, which has a little M, and there's transfer RNA. Messenger goes in there and reads off when it can access the chromatin. It goes in there and it unzips the double helix and it reads the sequence and it makes a copy of it. That's called transcription. Messenger RNA does that. Then it goes out in the nucleus to the ribosomes in the cytoplasm. Now remember what ribosomes do. They make proteins. And then transfer RNA goes in there and starts attaching the amino acids to the cytoplasm. How do they know to do that? We don't know. But that's not the mystery. The mystery is where's all the other instructions? Okay, transfer RNA reads the code on the messenger RNA, so the messenger RNA transcribes it, the transfer RNA translates it. And then the amino acids are correctly aligned to form peptides and proteins. And that's all they do, guys. DNA strand is copied onto messenger RNA transcription. Messenger RNA leaves nucleus, goes to the ribosome where proteins are manufactured. Transfer RNA base pairs with messenger RNA, which is called translation. Amino acids line up in the proper sequence along ribosome. Peptide bonds form. Bingo, we have protein chains. They terminate when all the amino acids are assembled in sequence. Okay. But trust me, guys, trust me, there's a lot missing. Here's a diagram of that. All right, we made it. I can appreciate this, the fact that what the DNA does, and it codes for these proteins and stuff. But what's missing is the shape of things, the morphology. How do we know where these things go? It's not in the DNA. It was thought that it's all in the DNA. It tells everything. No, it only tells a tiny little part. And the rest of it is a super mystery. Now, you can think about that while we move into the next one, but we're running out of time. Be with you next time. Be there, be square. Dr. Tom signing off. Hup.